Unit 6 is Gene Expression and Regulation. Genetic information is stored as a sequence of bases in DNA and RNA. Many viruses use RNA molecules to encode genetic information. Pyrimidians have a single ring structure while purids have a double ring structure. Purids usually have circular chromosomes, while eukaryotes usually have multiple linear chromosomes. Both of them could also contain plasmids. DNA replication happens before cell division. It is a semi-conservative model, which means that it results in a DNA molecule containing one original strand and a newly synthesized component. Each DNA strand has a terminal phosphate group on one end and a terminal hydroxyl group on the other end. The phosphate group is referred to as the 5' end, while the hydroxyl end is the 3'. The two strands run anti-parallel to each other, where the 5' end is opposite to the 3' end. Nucleotides can only be added to a growing strand in the 5'3" direction, so that means that there is a leading strand and a lagging strand. So in DNA replication, helicase unwinds the DNA strands. Polymerase releases the supercoil at the end of a replication. DNA polymerase then comes and synthesizes the new strands. It requires the RNA primers to initiate synthesis and attaches to the 3' end and builds strands in a 5 to 3 direction. Last but not least, ligase joins DNA fragments on the lagging strand. RNA molecules are used to facilitate protein synthesis. Ribosomes contain RNA and they assemble protein. During transition, DNA strands are separated. One strand serves as a non-coding or template strand, and the other strand is a non-template strand known as the coding strand. The gene targeted for transcription is located on the coding strand. The enzyme RNA polymerase synthesizes the messenger RNA into the 5 to 3 direction. It reads the template non-coding strand to the 3 to 5 direction. There are 3 types of RNA. Messenger RNA carries genetic information from DNA to the ribosomes. A codon is a 3-base sequence found on mRNA. Transfer RNA are recruited to the ribosomes to help create a specific polypeptide sequence. They carry a specific amino acid. Anticodon is a 3-base sequence on tRNA. The correct base pairing of tRNA anticodons with mRNA codons will result in the release and addition of amino acid to a growing polypeptide. Ribosomal RNA are a part of the ribosome, and they are just basically the functional units of it. In eukaryotes, after the mRNA is transcribed, an addition of a poly tail with like A. It helps with exporting from the nucleus. TP cap is also added. It protects the transcript and helps the ribosome attach to mRNA. After this, there's still a series of things that need to be done. Introns are sequences of mRNA transcript that do not code for amino acids. That means that they are removed. Exons, however, are sequences of mRNA transcript that do code for amino acids. So mature RNA only has the exons. This is called alternative splicing. This means that on the same gene, different mRNA transcripts can be produced. The first step of translation is initiation, where the rRNA in the ribosome interacts with the mRNA at the first start codon. The second step is elongation, where each newly arrived tRNA brings another amino acid to be added to the growing polypeptide chain. Then the last step is termination, where amino acids continue to be added until the stop codon is reached. Regulatory sequences of proteins are used to control transcription. Epigenetic changes can also affect gene expression. Histones are proteins that are used to wrap DNA around. Slight chemical modifications of DNA and histones cause tight packing or loose packing of DNA, and that regulates gene expression. All cells have operons, which are closely linked genes that produce a single mRNA molecule. It includes the genes to be transcribed, the regulatory sequence, and the operator. An operator is a sequence that either inhibits or promotes transcription by binding with regulatory proteins. In prokaryotes, these are transcribed by a single mRNA. In inducible systems, there's an operon that is inducible because it's usually turned off. There's a regulatory protein bound to the operator, which means that the RNA polymerase cannot bind to the regulatory sequence. Inducers are molecules that can bind to the regulatory protein, causing it to change shape. This causes the regulatory protein to release from the operator and frees up the RNA polymerase to transcribe the genes. The promoter region is where the initiation of transcription happens. All cells of a multicellular organism have the same gene. The phenotype of the cell is determined by the combination of the genes that are expressed. Mutations are the changes of a genome of an organism. Deletion and insertion shift the order of the gene sequence. Cells can go through transformation, which refers to the uptake of naked DNA. Transduction is the transmission of foreign DNA into a cell when the viral genome integrates with the host gene. Conjugation is the cell-to-cell transfer of DNA.